Good morning, children. This is Aniruddha, PGT Biology, Mount Litera School, Jhansi. In the previous session, we had finished chapter number 5, and now we are going to start chapter number 6, Molecular Basis of Inheritance. Inheritance and Variation chapter was chapter number 5, in which we talked about the Mendelian laws and other things. Now, in this particular chapter, we will be talking about the molecular basis of inheritance. And in molecular basis of inheritance, two things first of all come. Number one, it is DNA. Number one, it is DNA. And number second, RNA. Two things come, DNA and RNA. DNA is known as deoxyribonucleic acid and RNA is known as ribonucleic acid. DNA is known as deoxyribonucleic acid. Why? Because this is the structure of one nucleotide and the polynucleotide is DNA or RNA. And this is one nucleotide and its structure is, at the first position, this is ribose sugar. At the first position, one nitrogenous base is connected, and at the fifth position, one phosphate bond is there. So this is ribose sugar normally. So this is known as ribonucleic acid. But when ribose sugar becomes deoxyribose sugar, it becomes like this. This is the condition of one sugar, one nitrogenous base, one phosphate, and at the second position, the OH group which is present, the O has been eliminated through evolution. That's why this sugar is known as deoxyribose sugar instead of ribose sugar. This is known as deoxyribose sugar. And just because of this, the stability of DNA increases. DNA acts as genetic material in most of the organisms. RNA also acts as genetic material in some of the organisms such as viruses. But mostly RNA works as messenger in most of the organisms, and it also works as catalytic. It has one catalytic nature also. That is why RNA has dual function. It acts as genetic material also and it functions as catalyst also. So talking about the DNA. DNA is a long polymer of deoxyribonucleotides. What are these ribonucleotides? This is one sugar connected with one nitrogenous base. This is known as nucleoside, and when phosphate also joins it, it is known as nucleotide. This is one single unit of the entire DNA. When the same unit is repeated and they all are joined with one another, this is known as polynucleotide or polydeoxyribonucleotide chain. That is DNA. The total length of DNA in bacteria is 4.6 into 10 to the power 6 base pairs. And if we are talking about human beings, then it is 3.3, one single haploid content of human DNA. The human DNA is diploid. The human cell is diploid. That means a set of chromosome is found. So haploid set of chromosome contains 3.3 into 10 to the power 9 base pairs. So this is complete DNA. So DNA is made up of, first of all, I told you already, one sugar, that is ribose sugar, 5 carbon sugar, one phosphate is present, that is why it is known as acid, and nitrogenous bases are present. These nitrogenous bases are of two types. Number one is purine. Number second is pyrimidine. Purines are of two types again, adenine and guanine. And pyrimidines are also two types, cytosine and thymine. Purines have two ring structure, one hexagonal, one pentagonal ring, and pyrimidines have only one hexagonal ring. That's it. So this is the combination of DNA. Okay, this entire chart shows the same thing. So you can pause the video and you can see. The next point is, as I already told you, now Always remember one more point, that always adenine joins with thymine with double bond hydrogen, and cytosine always joins with guanine with the triple hydrogen bond. So this is always fixed, whether it is anything, any DNA of any organism. One more thing, mostly DNA contains thymine. In place of thymine in RNA, uracil comes uracil in place of thymine. RNA is a single stranded structure and DNA is double stranded structure. Okay, this is done.